We have again determinated by we have good right. So the last thing that was the is from demonstration. You from
Thank you, sir. So I won't take much of your time because I know I'm standing in the middle of your dinner time also. So I'm thankful to Dr. Masmi's presentation where she explained about the liquid biopsies, and one of the slides was explaining which are the different instruments. So I'm going to present one of the instruments from Applied Biosystem, which is called Quantstudio Absolute Q, which you can utilize for the liquid biopsy studies. I will be skipping some of the slides in the interest of the time. So when it comes to what is the difference between digital PCR as compared to qPCR, so what we do in digital PCR is that the reaction volume is compartmentalized in 20,000 microchambers. And each of these microchambers, they behave as a reactome. And if your mutant molecule is present there, then we get the fluorescence. And by comparing the fluorescence against the negative fluorescence, we can get the exact copies of the absolute quantification. This is the simplest mechanism of digital PCR. I just want to ask you one question. Can you tell me how many red candies are there in this particular jar? I understand it's really difficult for us to find out how many red candies are there. But suppose if I make compartments, and if I throw all these candies on those compartments, then we get something like this. Now, if I ask you the same question again, it is easier for you to count all the red candies. In fact, you can give us the exact estimations of those red candies. Some of the compartments might have one, or some of them might have two. Some might have a combination of candies. So what we do with the compartmentalization is we are reducing the noise and we are amplifying the signal. That's the most important thing. And compartmentalization is crucial when it comes to different digital PCR platforms. On the basis of techniques which are utilized for the compartmentalization, you have different offerings, and one of them is ours. Now, similarly, when we talk about your sample, especially in the context of oncology, let's say this is a 10 microliter reaction, and there are some 30 target molecules, mutants, against 30,000 wild types. So the abundance is 0.1%, and it's not possible to detect this particular molecules in the qPCR. But when we do the compartmentalization, we might something similar to this, that maybe one of the compartment might have one target molecule against two wild type molecules, and then we get the fluorescence. And it becomes easier because the abundance has increased to 33%. So that's the basic, principle of the digital PCR, where the enrichment is done by the compartmentalization. Now, when it comes to when exactly we should use it, when you want the accurate measurement of targets which are in low abundance, especially in case of oncology, when you're doing the monitoring, that's where you want the exact estimations of the mutant molecules or the resistant molecules for any of the therapies. When you are monitoring small changes over a period of time, maybe in the gene expression, when you have a low copies of transcript which are not detectable on qPCR, you can go with the digital PCR. When the absolute quantification is required, where the objective is to find out exact number of copies per microliter. Sometimes we want to do the abundance, from those samples. When we're asking the complex question, for example, CRISPR-Cas9, and in qPCR, the inhibitors, they might not give you the signals. So this is more, it is better than qPCR. The digital PCR is better than qPCR because of this compartmentalization, we can remove the inhibitors and we can get the signals. So these are the different areas where you can, put the digital PCR. Now, it doesn't mean that it's a new technology, it's been there. However, there are some inefficiencies of the existing platforms. For point number one, they have multiple instruments, so multiple hands-on time, which is tedious workflow. The time to result is long, because of multiple instruments. And the most important point is bed volume. I will talk about this later on, but they have wastage of reagents. And the last is inconsistent compartmentalization. If we go with the guidelines, you need to have 20,000 wild type molecules when you want to report 0.1% frequency of detection. So in order to address all these questions, we came out with Quantstudio Absolute Q. In terms of reagent efficiency, means how much you are loading and how much of that is going to participate in the reaction. More than 95% of your reactions are getting compartmentalized. it actually means that I'm going to get the complete picture. In terms of consistency, you get 20,000 microchambers, so you don't need to repeat the samples again because samples are precious. Easy to use, it's as simple as qPCR, just less than 5 minutes of hands-on time. It is 90 minutes, it's a fully automated platform, single instrument, which does everything. It is fully automated. It's a walk-through operation. You just start the run, go back and do something else, come back after 90 minutes, fastest time to the results. And another unique feature which we provide is auto false positive rejection. In terms of flexibility, you can run as long as 4 samples. So one of the wells can have your sample, second can have an NTC, third positive control, fourth negative control. So even if you have 1 patient sample, you can finish the same reporting the same day. And in terms of multiplexing, there are 4 optical channels available for the multiplexing. And the 5th optical channel is the ROX dye, which is utilized for the QC criteria. Now, as I mentioned, I focus more on the drug volume as auto false positive rejection as well as the flexibility. The picture on the left-hand side, the first picture, what does it show me? I have got a yellow car, and if somebody asks me whether the yellow car is present in this parking slot or not, if I have a complete picture, then I can say yes, it is there. But suppose if only 50% of the car park is visible to me, I will say there is no yellow car, but the fact is that there is a yellow car. Similarly, even at 30%, the probability is again very high that I will not be able to see the yellow car. Same thing happens with the sample. When you're talking about the monitoring, when the clones might be very low in number, right? So when you have limit, those kind of low mutant molecules, or resistant molecules, then what happens if you are not able to compartmentalize the majority of your sample, they don't participate in the reactions. In our case, more than 95% participate. So in this case, there are 3 mutant molecules. Probability is very high that all 3 of them are going to compartmentalize and they will be participating in the reaction. So you get 3 positives. But suppose, if the supplier X, they are providing you a system where the dead volume is almost more than 25%, which means how much you're loading and how much of that is going to participate. The probability is that probably one of the mutant molecule will be compartmentalized. So the fact is that there are 3 mutant molecules, not 1, but the report will be that, okay, there is only 1 mutant molecule. And same thing, if this increases further, then you may not get any kind of a mutant molecule in these kind of low sensitive, detections. So this is where the dead volume plays a very crucial role when we're talking about these kind of samples. Again, the background subtraction, digital PCR is highly sensitive technology. The sensitivity level of NGS as well as your qPCR is somewhere 1 to 2%. And digital PCR starts from there, goes up to 0.05% of the sensitivity level. So it's highly sensitive. So sometimes non-PCR artifacts can also give you signals. So what we do actually, we have a pre-PCR scan stage, this particular stage. Even before the PCR is done, we take an image. And actually the PCR is taking in the next stage, and then we take a post-PCR image. If we utilize algorithms and compare these images, we can remove the signals which are coming from the artifacts. Whereas in all the competitive platform, they take a post-PCR image. So suppose in 20s, and we're also going to give you the fluorescence that will be detected, and the probability of increasing false positive calls are very high. In reality, when the monitoring is done, and suppose if there are only 3 clones, so when you see these kind of results, and when it comes to whether I should report it or not, there is a question, is it really true or not? So at least by rejection of, false positives, you get more confidence on your data. So this is a unique feature which is present only in this particular platform. Now, when it comes to the workflow, the first step, as simple as qPCR, take the primer probes, master mix, DNA. Second step, you leave your sample on the plate. I'll show you the plate in the next slide. Go to the instrument, start the run, come back after 90 minutes, and you have your data ready with you. So less than 5 minutes of hands-on time with the full automated workflow. Now, as I mentioned, the plate, this is a plate. The technology is called microfluidic array plate technology. So we have a plate which is actually 4 arrays. These are the arrays, and each array has 20,480 fixed microchambers. Because we have fixed microchambers, we are not generating them. That's why the consistency is very high, that every time we get more than 99% microchamber fill success rate. More than 95% of your sample is analyzed. And the input volume is 9 microliter. It's very simple, you can go to the different vendor side, download the user guides. Everyone has got these micro chambers. If you know what is the total number of micro chamber and what is the volume of each of them, then you get the total volume and how much you're loading. Then you can find out, okay, what is that volume. So we use 9 microliter. The total volume of our plate is approximately 8.8 microliter. That's why we utilize more than 95% of the samples. This is a plate. Another unique feature, the plate is reusable. So minimum 4 items needs to be utilized, and remaining items can be reutilized again. So this is another unique feature available in the system. So it's a one system, fully automated, one plate, and we have the DNA and RNA master mixes available. As far as the software is concerned, all the features that are required for setting up the protocols are available. You can modify them also. You can upload the protocols also, and when you set up the run, it gives you the time also. Okay, this is the time that it is going to take for the completion of the run, which means you can go and do some other works at the same time. So when it comes to applications, these are the different applications. The first is important in case of oncology, it's rare target quantification or SNP discrimination. Second is absolute quantification when you want the exact number of copies, copy number variation. Another important application in oncology, gene expression. If you're looking for the low expressed transcripts, then digital PCR comes into picture. Even in the cell and gene therapy, we have complete solutions available for that. And disease and wastewater monitoring, for example, SARS-CoV-2. So we have a list of liquid biopsies assays available. More than 100 markers are already validated, which are available to you as a single plex assays. However, if you want to make the multiplex assays, if you want to get full benefit of the digital PCR by utilizing multiple optical channels and reducing the cost, then we can also work with you and develop those custom assays for you. We even have a custom design software available. Just a use case to give you an idea, I have taken an example of NSCLC. So we all know that patient comes to the clinician, he has got some symptoms, and clinician suggests for the biopsy. The preferred choice of instrument is NGS, so that we can get the complete profiling, and then we get the driver mutations, which actually means the patient is suffering from the NSCLC. Then starts the therapy, and this is the patient gets well, it goes back, and then there comes the time of monitoring, maybe 3 weeks or maybe 6 weeks, depends. When the monitoring happens, then you can take the 10 ml of blood, take the cell-free DNA or ctDNA out of it, and then you already know which are the markers for the first line or second line of therapies, and you can develop a panel to detect them. So every time you want to do the monitoring, you can take those particular samples on digital PCR. One example where you can understand how exactly the monitoring can be done. This is an example which we are going to launch as a product. So what we are doing actually, we are trying to bring more markers in the same assay, so your cost can be reduced. So you can have a panel like driver mutations, L858R can go to one of the dye, EGFR exon 19 deletion can go to the second dye, and we need the wild type because we also need to find out the minor allele frequency for all those mutations. Similarly, you can have a drug resistance marker T790M, C79S, 792, 718Q as well as wild type. Again, so we are utilizing all these optical channels so we can develop more and more multiplex panels. There are companies like ChromaCode. They are the platform agnostic companies. They have developed a panel of 14 EGFR DNA mutations and 15 fusions. They presented their data last year in the month of November in the AMP, and basically it was done on our platform. So those options are also available to the customers to utilize digital PCR. As far as single plex panels are concerned, we already have a list of almost 900 different markers. You can just select any of them and you can straight away go for them. Even if you want to do the multiplexing of them, you can let us know the assay IDs. This is the long list. I would suggest you can go to the website and look into all the new markers which are available. Similarly, you can also utilize digital PCR for the CNV analysis. In fact, with digital PCR, you get the exact estimations of the copy numbers. And as I mentioned, for the gene expression profiling also, because there are 4 optical channels, so you can utilize 4 different genes for the, especially the lowly expressed transcripts. I'll just skip them. So with this, I'll just come to the end of my presentation. So we have a single full integrated instrument, which is as simple as qPCR, takes 90 minutes to finish the run. And most important, more than 95% of your loaded sample is going to participate in the reaction. Consistently gives you 20,000, so there is no need for you to repeat the samples again. Flexibility, minimum 4 samples to 16 samples in a run of 90 minutes. It has 4 color capacity, and it can detect false positives, which gives you the confidence. So with this, I come to the end of my presentation. Any questions?